atmospheric CO2 has been increasing due to anthropogenic inputs. Ocean PCO2 has also been increasing with a concurrent drop of pH in the ocean. To understand this chemistry a little bit better visually, as we put CO2 in the ocean, it combines with HCO2 or H2O, water, and briefly forms carbonic acid before dissociating into bicarbonate and hydrogen ions. As we add more CO2 to the ocean, we have movement towards more bicarbonate ions and less carbonate ions. And this occurs because as we're adding hydrogen ions, the pH drops. And this favors the bicarbonate ion instead of the carbonate ion. So carbonate is important because when it combines with calcium, it forms calcium carbonate, which is obviously forms the hard shells and skeletons of many of the creatures down the ocean. And the mineral form of calcium carbonate that I'll be talking about today is aragonite. And aragonite is the form of calcium carbonate used by hard corals or reef-building corals. And so remember, as carbonate is decreasing in the ocean, so is aragonite. And while it's not really a direct correlation, the ease by which coral can calcify depends on the amount of aragonite or the aragonite saturation in the seawater. So at current, when we think of most coral reefs, we think of shallow reefs. And the aragonite saturation in shallow reefs is between 3 and 5. So that's way above aragonite saturation of 1. And we use 1 as kind of a general value to describe the saturation state in the water. So below 1, we would expect kinetically that the pure mineral of aragonite would dissolve. So in contrast to shallow water corals, when we move down to the deep sea, we actually have aragonite saturation closer to 0.6 to 3. And that's because aragonite saturation decreases with increasing pressure and decreasing temperature. The coral picture here is Lophelia pertusa. It's the most common cold water reef-forming coral in the world, and it's found in all the seas and all the temperate seas. They're also known to be an important source of habitat for many important commercial species. So I told you that we would expect aragonite to dissolve below a value of 1. So our first question, obviously, is how are they surviving below the value of 1? So it's important to remember, first of all, that all coral skeleton is covered by a thin layer of tissue. And so the skeleton itself is not necessarily experiencing the same conditions as seawater. Furthermore, corals can actually pump hydrogen ions out of their tissue, which increases the pH. And it's actually kind of like the opposite of ocean acidification. So as you are increasing the pH, you're favoring carbonate. And you can supersaturate your tissue in order to calcify the skeleton. Using boron isotopes, authors have recently shown that deep sea corals have the ability to pump these protons out even better than shallow water corals. So in this graph from 2012, the college shows that the change in pH between calcifying fluid of corals and seawater on the y-axis and seawater pH on the x-axis. And this graph is actually really powerful in that first that it shows the ability of corals to upregulate their pH in their tissues, but that upregulation is still tightly linked to seawater pH. And also, the colored circles, you'll see, are deep water corals. And so their ability to upregulate beyond the seawater pH is enhanced compared to shallow water corals. So from these kind of studies, along with calcification studies, we've seen that there's been a lot of papers recently discussing the resistance and the resilience and the acclimation of deep water corals. And Lophelia pertusa, the species I was showing earlier, has actually been shown as to be among the first to acclimate to long-term exposure to CO2. But most of my questions really kind of relate to the bigger scale questions and whether this kind of resistance, resilience, and acclimation is still going to hold when we think about the community and regional scale effects. So deep sea corals provide a great opportunity to answer some of these questions at a really large scale because Lophelia, as I said, is found all over the world. By comparing the quality of the habitat it provides in each of these regions with where we know the carbonate chemistry differs, we can gain a lot of insight of how all coral reefs will fare as the ocean acidifies globally. So here I've plotted a very general regional scale profiles of aragonite saturation with depth. So depth on the y-axis and aragonite saturation on the x-axis. And like I said, this is kind of regional scale profile, so averaging a lot of the carbonate chemistry that's available. And in the black shading is the depth distribution of Lophelia. The dotted line represents an aragonite saturation of 1, so below which pure aragonite would be kinetically favored to dissolve. And the point where that dotted line meets the profile is what we call the aragonite saturation horizon, or the depth at which aragonite saturation is equal to 1. You'll see that on these graphs of coral locations, the only place that occurs where corals occur below the saturation horizon is in the Southern California Bight, or the SCB, all the way on the left. So in the SCB, nearly half of all occurrences are below the aragonite saturation horizon of 1. The graph could be truly, this whole graph could be an entire presentation in itself, explaining why each profile is shaped the way that they are. But the reason the Southern California Bight is so acidic is because of upwelling of old DIC-rich and cold water. Factors that can contribute to the shape of these profiles generally coincide with upwelling, the age of the water mass, temperature, and how much freshwater input there is. And so that will, of course, come into effect as we move over to the South Atlantic Bight and the Gulf of Mexico. So while the Gulf of Mexico and the South Atlantic Bight approach 1, at these depths they don't actually reach 1, while the North Atlantic stays well above it. And the Mediterranean has very high dissolved inorganic carbon, but also high total alkalinity, which results in a high aragonite saturation state. So for this presentation, I'm actually going to exclude the Mediterranean from the deep water coral distribution data because it's anomalous in a lot of ways. One of them is that it's relatively less studied, but it's also really quite warm for many of the deep sea corals I'm talking about. So it has other reasons to be considered marginal habitat than the carbonate chemistry. So if you exclude the Mediterranean, what we kind of see is a pattern in which habitat quality actually tends to match these aragonite saturation profiles. So with the highest quality being where the average aragonite saturation over the distribution of the coral is highest in the North Atlantic, where we find Lophelia reefs that are kilometers across. And then this moves to South Atlantic Bight, where we also have high quality habitat, to the Gulf of Mexico, and finally to the Southern California Bight. So deep sea reef building corals were thought to be absent from the Southern California Bight and really the Northeast Pacific in general. But our research in the past few years has shown otherwise. So with that, I'm going to focus on this region first. So showing you what we've learned in the past 5 years or so. Prior to our research, there was less than a dozen records of Lophelia on the west coast, with the furthest north of which was reported by Jeff Hyland, who's sitting right next to me, in the Olympic Coast National Marine Sanctuary. And looking at just the Southern California Bight in general, we see we have about 7 historical occurrences. So we were given the opportunity by our colleagues at NIMS, including Kevin Stierhoff, John Butler, and Mary Yakovich, to review over 500 surveys they had done for rockfish and other species in the region in the past decade or so. And so with a lot of photo and video review, we were able to add over 100 new occurrences shown in yellow, with the white as absence and dives, and again with the red as historical occurrences. And Lophelia was present at over half the sites that we visited throughout the survey. And just in April, you'll see that star I added up to the north. We were all on a cruise in the Channel Islands National Marine Sanctuary where we added a whole other occurrence kind of at the northern bounds of our study area. So what, there's a lot of coral there, but what's important, as I said, is that we didn't really expect it there. So using long-term hydrographic data from the CalCOFI or the Cooperative Fishery Investigations, they have over 50 years of data, and we used an empirical model developed by Allen et al. 2012 based on temperature and oxygen to derive aragonite saturation. We also verified that these benthic temperatures and oxygens matched the hydrographic surveys using the CTD data collected from our ROVs. And carbonate chemistry was also collected directly by CalCOFI over the past couple of years and publicly available online. as well as by us on the sanctuary's vessel Shearwater and by the Belshimana in April. So using this data, we mapped average aragonite saturations at 4 depths. And you can see that spatial variability is relatively reduced at depth, going all the way down to aragonite saturation of 0.6 at 300 meters. This is among the lowest values ever reported for reef-building coral. And what's more is that these low aragonite saturation states are persistent. The maximum saturation at any single time point in the Calcofi database is lower than every reported value for 70% of our occurrences in the Bight. So aragonite saturation is really low. But as much as we wanted to think about these kind of points on a map, we wanted to go further and think about what these points represent. Because Lophelia in the Southern California Bight doesn't look like this. This is an example from the Gulf of Mexico. Lophelia in the Southern California Bight looks a little bit more like this. And 84% of all occurrences were only small or sparse aggregations or rubble. But interestingly, overall abundance of coral actually peaked at our deepest site in the Channel Islands National Marine Sanctuary called Piggy Bank. So it's a little counterintuitive because that's also where aragonite saturation is the lowest. But there was an opposite pattern kind of in reef cohesiveness, as I call it, or density of the aggregations. And these only occurred at the shallowest sites where aragonite saturation averaged over 0.9. So in analysis of the whole Bight, taking into account coral health, we see that cumulative live coral exceeded dead only at depths shallower than 169 meters or above the aragonite saturation state of 0.9. In the shallowest third of the depth range, coral cover was 60% live, dropping to near 30% for the deepest 2/3 of the depth range. But if you take a look at the graph, you can see that cumulative coral does not increase smoothly across depths, but rather has these large jumps in the cover. And that's because a lot of these were based off very notable occurrences. So while it was widespread, there were certain occurrences that were driving all the statistics. To evaluate the pattern and density and kind of reef cohesiveness, we looked at, we gave values of formation of framework, with 1 representing cohesive framework to 3 of only isolated patches of rubble. And we found significant differences in the carbonate chemistry between categories, with only category 1 having an overall mean aragonite saturation above 1. There was only 1 framework occurrence in all of the Bight below an aragonite saturation of 1. But unfortunately, covariation in these carbonate chemistry parameters somewhat prohibits identifying any one of these as controlling factors. But we do see a clear pattern emerging of an increase in patchiness with a loss of reef cohesion with an increase in depth. But like I said in the beginning, this doesn't necessarily correspond to the abundance at sites. So we have to look at it in multiple ways to kind of get a full picture. So even this still doesn't give us a full view of how the coral is doing in the Bight. So recently we've been working more at kind of the organismal level. And what you can see here are examples of Gulf of Mexico coral on the left and Channel Islands National Marine Sanctuary coral on the right. So the two are 2 different forms of morphotypes that we see in the Gulf, but they do co-occur together. And clearly there's a big difference. Both in the linear extension, we see profuse budding in the Channel Islands coral, and we also see a corresponding reduced skeletal density. And this 2.8 is really what we see both in the Gulf and the North Atlantic. So we see a pretty big reduction in skeletal density. But even further, we did some SEM work thanks to Andrew Schuler, who also works in our lab here. And we've been able to look at the skeletal material comparing Gulf of Mexico Lophelia, which occurs at aragonite saturations of around 1.5, to Channel Islands, where we have collected around 0.7. And preliminary evidence from the outer skeleton of this Lophelia, where we see these bumps, shows that we have reduced vertical extent of the bumps on the right with low aragonite saturation states, as well as kind of an irregular crystal structure. It's a little hard to see from here, but they're less tightly linked from those samples that we see in the Gulf of Mexico. So this is really just preliminary work. We've done a couple to try to get to notice the big differences that we see so far. So moving up to the helices, We had some problems with broken septa, but you can still see that the bumps along the septa exhibit a far less organized pattern here on the Channel Island samples compared to the more concentric rings that we see in the Gulf of Mexico. My best guess is that this has something to do with growth patterns, and that maybe the Gulf of Mexico is more time-based growth, where Channel Islands might be food-based growth, but we don't know for sure. And if we look at the very ridge of those septa, we can also see that the bumps are only present in the Gulf of Mexico, and there were no bumps on the outer ridge of the Channel Islands, perhaps indicating that there was current growth at collection for the Gulf of Mexico samples. But one of the clearest signals we saw of the corrosive water in the Channel Islands were these big dissolution spots that occurred on almost every sample we collected. So you can actually see this visually, obviously without the SEM on the right, just exhibited as kind of a chalky appearance. And when we looked with the SEM, it became clear that these were all areas of dissolved aragonite crystals. So with these non-tissue covered sections in mind, we wanted to think a little further about how these contribute to the reef. And interestingly, dead reef matrix arguably contributes more to the habitat than live coral. In addition to adding continued growth in terms of vertical extent, dead reef also supports far more species apparently than live coral because live tissue prohibits settling of epibionts. But furthermore, skeletal holdfasts at the regions of the base of coral are not covered by tissue. They cement the coral to the substrate, and their integrity is vital for the coral's ability to maintain their position on the rocks and boulders that extend into the water column. And in the Southern California Bight, we always see these kind of habitats in very high relief areas on these rocks and boulders. So if they can't hold to them, what we often see is a lot of rubble on the bottom with very little live coral on top. So furthermore, inorganic cements are often seen in shallow water settings to help bind coral together in supersaturated conditions. This precludes porosity and will help the overall reef integrity. And because these are in low saturation conditions, we don't have that either. So just to briefly touch on these little bottle experiments I have under skeletal holdfast with dead reef matrix, that was part of my thesis. And part of the reason that we know that what happens is they slowly dissolve below 1, but then that becomes more rapid at around 0.8. And this differs from the value of 1 I was telling you before, because pure aragonite, coral aragonite, has various proteins incorporated and alterations to the mineral structure by coral. So we know that they don't dissolve exactly at 1, which kind of changes a bit of the framework and when we're thinking about this. But so going back to the overall hypothesis of resilience, we can see that beyond calcification and the boron isotope work, which is obviously important, we might not necessarily see the community level resilience based on the dissolution of kind of the backbone components that hold the reef together. Species-wide in the Bight, Lophelia, I didn't really have a good figure for this, but Lophelia is likely able to persist because of a rich food supply. So a recent paper oddly found that phytoplankton was among the most efficient sources for conversion to fatty acids. And that suggests an important role in shallow water, cold water reef settings. So this would be the case in Southern California, where we see Lophelia much more shallow than other places in the world. So in Southern California Bight, subsurface chlorophyll maximums are near depths of Lophelia that suggest the potential role for enhanced food supply. So the importance of food supply is further supported by the locations of our 2 deepest notable occurrences. And these are both found in highly dynamic regions of eddy formation and upwelling. So despite mean aragonite saturation below 0.8 at 200 meters, we think that Lophelia pertusa is probably able to calcify based on their food supply. So there'll be a balance with maintaining sufficient energy requirements at locations of enhanced food supply. So even though we know they can upregulate, we know that it probably comes at a cost. So looking into the past, present, and future of the region, we can see that in pre-industrial times, seawater was saturated with respect to aragonite at all depths of Lophelia locations, and now only occurring at about half the time in saturated conditions in the middle graph. And by 2050, all aggregations in the Southern California Bight are expected to be in seawater that favors their dissolution. Another important way of thinking about this is in terms of an organism's climatic variability envelope. And so while estuarine organisms may experience a relatively large envelope of conditions because they experience a wide range across both daily and weekly and seasonal scales, deep sea organisms only see a relatively narrow range. So this makes the Southern California Bight a region especially important to the OA scene because the SCB is not only approaching aragonite undersaturation, so this threshold of 1, but it's also moving outside its present-day envelope of conditions with only a couple of decades. So within our lifetime, we can understand both our lifetime and the lifetime of coral, what may happen pending kind of the future ocean acidification scenarios. So from a regional to the SCM level, we've kind of used the Southern California Bight as a natural laboratory for ocean acidification. We were able to use available data, both in terms of obtaining the coral records and hydrographic data, to think about the big picture idea of how coral reefs will fare globally if the ocean acidifies. We also gave the first description of a widespread reef-building coral on the west coast, showing that while it's widespread, framework extent, linear extension, and skeletal densities were all reduced relative to other regions in the world. So in some ways, it's encouraging to know that survival of species can occur in these corrosive conditions, and it does coincide nicely with some of the laboratory work that's been shown. We can't really ignore the functional loss of the species at the reef framework and community level. So I just wanted to move to our coast now and introduce kind of a similar premise in using available data to move towards a better understanding of OA effects in this region as well. So the white and black dots here represent GOMEC data, and this data was presented by Rick Winnenkoff a few seminars ago, collected in both 2007 and 2012. Data was also collected and presented in a few papers from 2005 and 2006. So there's a good amount of hydrographic surveys that have started to go on in the area. And the white, orange, and pink dots represent deep sea reef-building corals from NOAA's National Database for Deep Sea Corals and Sponges that is currently being built by the Deep Sea Coral Research and Technology Program and Robert McGuinn, who's also in the lab here in Charleston. So you can see that not only are these corals extremely abundant in the southeast, but they also overlay the carbonate chemistry stations that have already been collecting data. And briefly, before moving further into the southeast, I wanted to move north and give you a regional perspective that the abundance of deep reef builders drops significantly as we move north. Robert actually reiterated to me today that this is partially due to a lack of effort and data that's made its way into the database yet. But we also know from papers like the State of Deep Coral Ecosystems that the North does not appear to have an abundance of large structure-forming deep corals and deep coral habitats. So I included a graph on the left from also what was presented a couple seminars ago in the Winnenkoff et al. 2015, showing aragonite saturations at depth. And it shows an overall pattern of a reduced aragonite saturations as we move north. So while we can't say it's the single culprit for the lack of reef builders in the north, I do think that it probably has at least some sort of contributing component. A recent cruise by Brooks et al. In the Mid-Atlantic canyons found among the first aggregations of Lophelia there, but they were also small and never reef building. So they suggested that turbidity in the canyons or limited larval display, limited larval supply could play a role in that. But like I said, I think the overall patterns of reduced aragonite saturation states do kind of support the lack of reef builders that we see up there. Moving back into the the southeast, I just wanted to go into one of the transects that they also presented in the same paper. So like I said, we haven't collected our own data yet. We're just kind of put the pieces together. So this is a transect along the Florida Straits. And the white and the orange lines along the bottom represent the distribution of Lophelia and white and oculina in this pretty immediate vicinity, not exactly along the track, but close enough. And the aragonite saturation data shows that the coral is persisting in relatively low aragonite saturation, so below 1.5 as the saturation stage shoals moves towards the coast. And this actually shows this line of 1.5 shows all the way up to 75 meters, so that does also incorporate a lot of the oculina banks that we see in the region. So while widely distributed in this region, the other thing that makes it a lot different than Southern California is that they form substantial reef-like structures that we don't see in the West Coast. So we see live coral sitting up atop really large dead coral mounds. And given the vulnerability to low aragonite saturations that we showed on the West Coast, we can likely say that these corals in the southeast will eventually be affected by impending ocean acidification. And furthermore, the importance of dead reef matrix these habitats might render them especially vulnerable given the susceptibility of non-tissue covered portions to the corrosive waters. And like I said, these are building on large dead reef mounds. But what we still have yet to understand is kind of the climate envelope with which they exist. Because the research shown between 2007 and 2012 shows that there's more variability in aragonite saturations than from non-anthropogenic causes than from the signal that we would actually expect from ocean acidification. So this has to do with changes in currents, changes in temperature, salinity, freshwater inputs, and other changes outside what we can measure on a 5-year scale with ocean acidification. So what's important will certainly be to help define the envelope of the corals in this region. So thinking about what's next for the southeast, I think there's a lot of potential for regional scale modeling like we did in the Southern California Bight because of the large depth range of Lophelia and wide distribution. And in a way, I think the East Coast may be even better suited for it because all the other environmental parameters with which we know Lophelia exists throughout the world actually match on the East Coast a little bit better than the West Coast. I don't have time to go into all the different parameters of Lophelia that we see on the West Coast, but we do know that while temperature is about right, oxygen is pretty low, so that contributes to the patterns we see as well on the West Coast. But on the East Coast, we know that there's suitable habitat, the oxygen is generally right, and that and temperature as well. So if we eliminate some of those variables, we might be able to see a better pattern at the regional scale of how aragonite saturation might affect these reefs. And I think to some extent this is underway by Steve Ross, Andrew Brook, and some of the other colleagues that have started collecting water closer to these coral aggregations. So looking at what I put up there, the first thing obviously is to move beyond points, because I think our West Coast story evolved quite a bit when we thought about how many occurrences were there in such an acidic environment, but then when we took a little bit of a closer look to think about kind of the habitat quality they provide. So the Southeast might be among the best regions to do that where available data exists for a lot of these reef habitats. Secondly, we'll need to continue kind of the benthic characterization of the carbonate chemistry. There's a good chance this does differ from hydrographic surveys, and we'll need to know to what extent they differ and whether we can kind of rely on future hydrographic surveys to have an understanding of how the benthic habitats are changing. And lastly, and most importantly, probably setting up long-term monitoring. We're fortunate to have marked colonies in this past April in the Channel Islands Sanctuary, but in a way we're kind of looking at communities already on the brink there. So in the Southeast we really have a potential better way to get a baseline data of reef-building corals in the area. So in a large scale, I think we're beginning to have really nice data that complements a lot of hypotheses that were thrown out a decade or so ago by like Renaud et al. And others, suggesting that deep sea coral reefs are really limited globally by the aragonite saturation horizon. And the shallowest horizon is found on the west coast, shoaling as you move from Southern California up to Washington. And the distribution of deep sea corals on the west coast also matches this, with an increased occurrences as you move south, but still limited in condition and extent in the corrosive waters. Aside from potentially limits to research effort, this pattern also shows to be potentially occurring on the east coast as well, with the highest abundance of corals in the saturated waters of our southeast. So as the ash is shoaling globally, with potential shoaling by as much as 2,000 meters in the North Atlantic, this is where most of the deep water reefs are formed. So conditions are changing rapidly. And it's expected that by the end of the century, about 70% of all known deep reef-building corals will be in undersaturated conditions. And with our evidence from the West Coast, we know that this could have some significant dire consequences for the reef-building corals. So I just wanted to go back, since this is kind of a Southeast seminar, thinking about what we know and where to go. As I said, the Southeast has the highest abundance of deep-sea reef-building corals in the U.S. And these corals are probably at aragonite saturation a little bit lower than we might have thought in the beginning because of those shoaling aragonite saturation profiles as you move along the benthic boundary layer. So we have a great start with the carbonate chemistry, but of course would benefit from much larger scale, both at a spatial scale to capture all the occurrences that we have, as well as a better understanding of the temporal extent. And what we have shown thus far is that occurrences are likely occurring at aragonite saturation below about 1.5, so still pretty low in the grand scheme of things, and much lower than shallow water reefs. And lastly, as I said, I think monitoring is extremely important. Now's the time to establish baseline and track the changes. And the Southeast really gives us a great opportunity to do that, where we have good habitat still. So with that, I just wanted to acknowledge in the upper right corner Dr. Peter Etnoyer and Dr. Jeff Hyland for their endless support in all this research, and the Deep Sea Coral Ecology Lab at SECOORA, especially Andrew Schuler, who helped with the SEM and on our past cruise, and Ren Salgado, who is also on the cruises as well and has greatly contributed to all the data that's gone into the West Coast, and Robert McGuinn, who helped me with his newly formed database to get all those occurrences in the Southeast. A lot of the data came from NIMS, especially Kevin Steerhoff and John Butler, Mary Yakovich, and also all the cruise support with Marine Applied Research and Exploration. That's Dirk Rosen, Andy, Rick, and Steve. So with that, I think I went pretty fast, but I'd be happy to take your questions.
Thank you so much, Leslie, for that very informative and great presentation. We're going to open the floor now for any questions, and we have a few that have been posted into the question box. So please continue to enter these questions, and we will pass them over to Leslie. So Leslie, our first question comes from Evan, and the question reads, could there be a link between photosynthesis in shallow corals or lack thereof in deep water corals, and how they handle high PCO2?
Yeah, there's definitely one of the benefits of studying deep sea corals, and I think that's why a lot of the laboratory research is actually started there in many ways, because we can untangle that pretty easily. So I would say most so because of the nutritional input that shallow water corals have, but that's kind of traded off with the fact that we know that deep sea corals have this ability to upregulate even more so than shallow water corals. So they're definitely not apples to apples, but I'm sure the ability for shallow water corals to photosynthesize will help with nutrition, and that's going to be kind of the main counterbalance to how corals are going to be dealing with ocean acidification. So the more nutrition you have, they've shown the better off you could be.
Okay, great, thank you. Next question, I think also comes from Evan. He wants to know, have you looked at differences in food availability between corals in the Channel Islands and the Gulf of Mexico, in addition to differences in saturation state? Oh, wait.
No, we haven't done that a ton yet, but we do know that the Gulf of Mexico has a lot of food as well. I think the food questions really get at this organismal growth level type thing, and the ability to survive and persist. And a lot of the questions we've been asking are more kind of at the mineral scale with how a coral can actually build a reef. But we haven't done that yet, and I think that would be a really cool way to kind of move next into thinking how each of these reefs are able to persist. There have been, I think actually at USC, they've been putting down some sediment traps in the Channel Islands. So apparently they were chock full. So we know from that, along with kind of the regional scale stuff, that there's a lot of food going down there, and that's probably our best, definitely why we think they're surviving. But we don't have necessarily good comparison data yet, but I know that there have been sediment traps from some of the Gulf of Mexico reefs, so I'm sure that's something that we can compare for sure.
We also know there's large aggregations and kind of high particle load environments.
I think you want to. Peter's on. Peter's next to me too, so he's trying to contribute to the questions.
Okay,
so
great.
Let me know if you can hear him.
Evan's asking a very interesting question. The Gulf of Mexico is an excellent place to study that because there's large aggregations of Lophelia pertusa close to the Mississippi River, where we expect high particle loads and high nutrient inputs, and then also off West Florida, more in the loop current regime, where we'd expect low particle loads and low nutrient inputs. So Gulf of Mexico is a good place to examine that contrast.
Yeah, it's a good question and a good way to move from here.
Okay, great, thank you. Lisa Robbins asked, were the corals in the SEMs from the Channel Islands living when they were collected?
They were living, yeah. That was what kind of surprised us in seeing all those dissolution spots. So we would know that a lot of the dead corals we would expect where the tissue's gone, we would see those dissolution areas. What we're actually seeing is that the tissue is sitting just on top of the polyps, and it's almost like the cenozoarch, which is the tissue that attaches all the polyps together and covers the skeleton, is missing in a lot of these places. We can probably look at this a little bit better. We actually have a lot of Lophelia in our tank now that Ren Delgado, that I list up there in the acknowledgements, has been taking care of. But basically from all of the, it's always mixed in together, but we do know that a lot of the live is still showing those dissolution spots in the SEM. So I think I went in deeper than, I don't know, I was assuming what you were asking with that, but yeah, a lot of them, the samples were alive when collected.
Okay, all right, thank you. All right, are there any more questions from anyone? Okay, all right, so I need to get the presentation changed back over to me for 1 second. So again, Leslie and Peter and Jeff, thank you so much for that presentation. That was excellent. If you think of questions that you'd like to pose to Leslie later on, please feel free to send them to us, and at the address that you see in the first line on this next slide, and we will be happy to pass those on to Leslie, and she'll get back with you with an answer. If you're interested in joining the SOCAN listserv, you can find the listserv address here in the center of the screen, excuse me, at the bottom of the screen. And if you have any direct questions about the SOCAN network, you can send those to us at SECOORA.org/SOCAN, and that's the bottom of the screen. So thank you for attending the SOCAN State of the Science webinar today. We welcome any feedback or further questions or suggestions for topics for this webinar series. You can submit input by replying to a follow-up email that you'll be receiving shortly. And the recording of this webinar and a PDF of the presentation will be available on the SOCAN website. Our webinar schedule is changing due to SECOORA's annual meeting that will be held in 2 weeks from today. So our next State of the Science conversation will take place a month from today on Tuesday, June the 2nd. And Lou Burnett, also from the College of Charleston, will discuss oceans acidic and low in oxygen, lessons from estuarine organisms at 12 p.m. Eastern Standard Time. We hope you'll join us for that webinar. So thank you again, Leslie, for your very informative presentation, and to all of you for your thoughtful questions and for taking the time to join us. We hope to see you at SOCAN's next State of the Science conversation. This is the end of the webinar session.